The ability of all living organisms to respond and react to external signals presents an essential component of life. One of the mechanisms used to transmit extracellular signal is the JAK3 pathway. The pathway is fairly simple and straightforward. Essentially, it all starts with a signal. In this case, it's usually a cytokine, a molecule produced by neighboring cells. This molecule will bind to a receptor on the cell surface, which will lead to a structural change. A protein which is bound to the receptor inside the cell, a Janus kinase, will get activated and express its function in activating other molecules through phosphorylation. It's going to activate the STAT protein, known as a transcription activator. The activated STAT protein will go into the nucleus, bind to DNA, and enable transcription of genes needed for cell proliferation, growth, and differentiation. The pathway works like a switch that can turn gene transcription on or off, driven by binding of the signaling molecule to the receptor. But what happens if one of the key elements of the system is broken? What if it's mutated in a way that it doesn't need a signaling molecule to get activated? Then the switch is always turned on, with no way to turn it off. This will lead to uncontrolled cell growth and proliferation, resulting in cancer. If it happens in blood cells, it leads to leukemia. For my project, I have worked on activating mutations of Janus kinase 3 that is present in patients with leukemia. To investigate it in detail, the mutation was introduced into a mouse model. Now, since this mutation exists already in patients with different types of leukemia, we expected the mouse to also get it, which surprisingly wasn't the case. The mice were happy and healthy. Why? Well, every gene in our body, as well as in mice, exists in two copies, two alleles. Sometimes one can get mutated, sometimes both of them. A mutated gene leads to a dysfunctional protein. So why did our mice with a dysfunctional protein not get sick? We assume that this is because the mice still had the non-mutated allele, which got transcribed and then translated into a normally functioning protein, which through experiments we have found to be more stable than the mutated protein. This is also in line with the patient data, where we have discovered that most patients with leukemia that have this mutation also have additional mutations or completely lack their normally functioning protein, making it easier for them to develop this type of cancer. These results help us understand the role that different mutations have on leukemia development. But why is it important? Leukemia arises faster than any other type of cancer. It is the most common cancer in children, and since it is not a solid tumor like breast or skin cancer, it can't just be cut out from the body surgically. But drug treatment or chemotherapies are necessary, and these usually cause severe side effects. So this kind of research is the first step towards developing safer and more efficient treatments for leukemia.